Harvesting the Sun, Part 2. In this part of the podcast, we will look at the light-independent reactions and how they use the ATP and NADPH produced as a result of the light reactions. This is a picture of Melvin Calvin doing experiments with radioactive CO2. These days he'd be in big trouble for not wearing a lab coat. Calvin, together with his co-workers Bassam and Benson, discovered the pathway for carbon dioxide fixation, which now bears his name. The Calvin cycle uses ATP and NADPH produced by the light reactions, but the CO2 fixation reactions themselves are light independent. You can read Melvin Calvin's account of the elucidation of the pathway of CO2 fixation that now bears his name in his Nobel Prize winner's lecture. This slide shows a photograph of the so-called Lollipop Apparatus used by Calvin et al. Radioactively labelled 14CO2 was fed to illuminated algae. Samples were collected after short time intervals. The incorporation of the 14C label into metabolites was detected, and the order in which the compounds became labelled was determined. The first compound that the C14 was made into was 3-phosphoglycerate, 3-PG. A lot of detective work went into sorting out which compounds were made and the order in which they were produced. This slide provides an overview of the Calvin cycle. Note the three phases: carboxylation, reduction and regeneration, and the use of ATP and NADPH formed during electron transport. The first step, carboxylation, is carried out by an enzyme called rubisco. This is the most abundant protein on Earth. Rubisco takes one molecule of CO2 and adds it to a 5-carbon acceptor molecule, ribulose bisphosphate, to make a 6-carbon molecule that rapidly splits to 2 molecules of 3-phosphoglyceric acid. When this has happened 3 times, we have a total of 6 3-PGA molecules. These molecules are phosphorylated using ATP and reduced using NADPH to produce 6 molecules of glyceraldehyde 3-phosphate, GAP, which is a triose phosphate. One of these molecules of GAP can be taken to make carbohydrate, and the remaining 5 are recombined to regenerate 3 molecules of ribulose bisphosphate needed for the cycle to continue. The regeneration phase is complex. 2 of the 3 carbons are combined to form a 6-carbon compound. A further 3 is used to give a 5 plus 4. The 4 then combines another 3 to give 7, and the 7 combines with the final, the 5th carbon, to give 10, which is then split to 2 5. So overall, 5 3-carbons become 3 5-carbon molecules. The GAP, triose phosphate, is exported from the chloroplast to the cytosol in exchange for phosphate, which is needed for ATP synthesis. Once in the cytoplasm, it can be converted in steps to glucose and then to sucrose. It's important to understand the relationship between photosynthesis and respiration. Photosynthesis, as we have seen, uses sunlight to power synthesis of ATP and carbohydrate. Respiration is the oxidation of carbohydrate to CO2 and water. Although the opposite in outcomes, they employ common mechanisms involving reduction-oxidation reactions and proton gradients to energize the synthesis of ATP. The terminal stages of aerobic, that is oxygen-consuming respiration, take place in mitochondria. Plants have mitochondria and respire just as animals do. This is essential for ATP synthesis in the absence of light, as well as production of many important biosynthetic intermediates. Plants also carry out photorespiration. This is a distinct process from mitochondrial respiration, although it results in the consumption of oxygen and release of CO2 in the light. Photorespiration occurs because rubisco, the enzyme that fixes CO2, can also incorporate oxygen into ribulose bisphosphate. Instead of 2 times 3-PGA, 1 times 3-PGA and 2 2-carbon phosphoglycolate are produced. Photorespiration is the process by which phosphoglycolate is returned to the Calvin cycle. This slide shows a simplified scheme of photorespiration. It's very complicated, involving 3 separate organelles: chloroplasts, peroxisomes and mitochondria. This elaborate pathway serves to salvage the 2-carbon phosphoglycolate that is split from ribulose bisphosphate when oxygen instead of CO2 is incorporated. It's 75% efficient. 1 out of 4 of the carbons is lost to CO2 during the salvage of 2 molecules of phosphoglycolate. So is photorespiration a bad thing? It results in loss of fixed carbon, and ammonia is also lost, and ATP and NADH, another form of reducing power similar to NADPH, are used up. When light energy absorbed by the photosynthetic apparatus exceeds the capacity of the Calvin cycle to use the ATP and NADPH, all the electron acceptors become highly reduced and the photosynthetic apparatus can become damaged, a process known as photoinhibition. Photorespiration uses up ATP and releases CO2, so it has a protective role. Gases and water can pass into and out of leaves via pores called stomata on the lower surface. They can be open and closed according to water availability. Under dry conditions, stomata close to limit water loss, but this also limits entry of CO2 and therefore favours photorespiration. Some plants, especially those that grow in hot, dry climates, have eliminated photorespiration by separating in space CO2 fixation and the Rubisco reaction of the Calvin cycle. CO2 is fixed by combination with a 3-carbon acceptor molecule to make a 4-carbon compound, hence the name C4 plants. It's transported into the cells that contain the Rubisco and the CO2 is released there. This means that Rubisco always has enough CO2 even when stomata are closed. Because of this adaptation, C4 plants are much more efficient at photosynthesis at low CO2. However, as CO2 levels rise, C3 plants should become more efficient due to reduced photorespiration. However, C4 plants are far more efficient at higher temperatures, so there's a lot of interest in engineering C4 metabolism into C3 plants to cope with global warming. So in summary, carbon dioxide is fixed by Rubisco, incorporating it into 5-carbon ribulose bisphosphate to give 2 molecules of 3-PGA. ATP and NADPH made by the light-dependent reactions are used to phosphorylate and reduce 3-PGA to GAP. 3 molecules of CO2 give rise to 1 molecule net of GAP. The other 5 are recycled to regenerate 3 molecules of ribulose bisphosphate. Plants carry out both photosynthesis and respiration. Additionally, under low levels of CO2, plants photorespire due to the ability of Rubisco to incorporate oxygen into ribulose bisphosphate in place of carbon dioxide. C4 plants have eliminated photorespiration by separating the initial CO2 capture and the Rubisco reaction. This helps them to photosynthesize at low CO2 levels when stomata are closed.